Hi, my name is Emily Hartso, and my project is using 3D printing to explain the pathogenesis of sickle cell disease. As a biology major and a digital culture minor, I wanted to combine the two disciplines in a final capstone project. The goal of my project was to teach a biological concept, in this case sickle cell disease, using knowledge I've gained in digital culture, such as animation and 3D printing. Here is the animation I made to explain sickle cell disease. In order to explain the pathogenesis of sickle cell disease in an easy to understand manner, I learned how to render and animate in Maya. Here you can see red blood cells flowing throughout your bloodstream. If you zoom in, you can see hemoglobin as it normally floats around your red blood cells, binding oxygen like it's supposed to. However, in sickle cell disease, there is a single base pair mutation which completely changes the structure of hemoglobin, preventing it from binding oxygen effectively and forming these long fibers. This extrudes the shape of the red blood cell, making it have its characteristic sickle-like shape and causing disseminated disease. In order to make my models, I had to download the DNA sequence from a website called Protein Data Bank. I downloaded the DNA sequences of normal and sickle cell hemoglobin and imported them into a program called PyMole. This program will automatically generate the 3D structures of the proteins, and these are the structures I used to make my 3D printed models. These models were converted to STL files and sent to the MakerBot printers in the Digital Culture Fabrication Lab. I used the MakerBot Replicator 2 in order to print my models. This is a fused deposition modeling printer, meaning that it uses additive printing of heating and extruding PLA filaments in order to form a scaffold on which the protein is being made. I decided to create these models to be 3 by 3 inches in order to easily fit in one's hand so people can easily interact with my proteins. Here are what the models look like right after printing. They are this natural white color, and after breaking off the scaffolding, you can see a lot of the detail. I also decided to paint the mutated model red in order to differentiate it from the normal model even further and also smooth it out. Overall, I'm very pleased with how my models turned out, and I believe that they, as a teaching tool, are very good in an accompaniment with my animation explaining sickle cell disease.